Hi guys, welcome back. Your fat is not just sitting there. Right now, the fat tissue in your body is producing hormones, secreting chemical signals, sending messages across your bloodstream, crossing your blood-brain barrier, and actively influencing how your brain regulates hunger, mood, stress, memory, and even your risk of cognitive decline. Your fat is quite literally talking to your brain, and a review published in May 2026 in the journal Obesities confirmed something that would have been considered radical science just 2 decades ago. Adipose tissue, what we simply call fat, is now classified as a fully functioning endocrine organ, not just a passive energy store, an active communicating endocrine system with dedicated hormones, dedicated signaling pathways, and documented effects on brain function at every level. In the next few minutes, you are going to discover exactly what hormones your fat tissue is producing and sending to your brain right now. The 2025 Nature Metabolism discovery showing that your fat cells send a stress and anxiety signal to your brain during acute psychological pressure. Why leptin resistance, a condition affecting millions of people, means your brain stops receiving the signal telling it you have eaten enough. How adiponectin, produced by your fat cells, directly influences neurogenesis and synaptic plasticity in your hippocampus, and what the latest research says about the adipose brain axis as a predictor of cognitive decline. Let's get into it. For most of medical history, fat was understood as a storage system. Your body took in more energy than it needed, and the excess was packaged into fat cells called adipocytes for later use. Passive, inert, waiting. That understanding began to shift in 1994 with one of the most significant discoveries in metabolic science, the identification of leptin. Leptin is a hormone produced almost exclusively by fat cells, and its primary purpose is to communicate directly with the brain, specifically with the hypothalamus, the brain's master regulator of hunger, energy, and body weight. When your fat stores are full, leptin levels rise. Those leptin molecules travel through the bloodstream, cross the blood-brain barrier, and bind to receptors in the hypothalamus. The message they carry is precise. Energy stores are sufficient. Reduce food intake, increase energy expenditure. This was the first documented proof that fat tissue was not just a storage depot, but a signaling organ capable of producing a hormone that directly regulated brain function and behavior. But here's what almost nobody knows about what the research has revealed since then. Leptin was only the beginning. Scientists have now identified dozens of signaling molecules produced by fat tissue, collectively called adipokines, each with distinct effects on different organ systems, including the brain. The 2026 review in obesity specifically identified leptin, adiponectin, resistin, and irisin as having documented roles in modulating synaptic plasticity, neurogenesis, and neuronal metabolism in the brain. Your fat is not sending one message, it is sending dozens simultaneously every hour of every day. But here's where most people get this completely wrong. Most people assume that more fat means more helpful signals, that larger fat stores simply mean stronger leptin signals telling the brain to stop eating. This is precisely backwards from what the research consistently shows. In obesity, as when fat stores are substantially elevated, leptin levels are also elevated chronically. The brain is receiving constant high-volume leptin signals day after day without interruption, and the brain responds to this chronic excess the same way it responds to any chronic overstimulation. It downregulates its own sensitivity. Leptin receptors in the hypothalamus reduce in number and sensitivity. The brain becomes unable to respond appropriately to the signal it is receiving. This is leptin resistance, and it creates one of the most frustrating paradoxes in obesity research. A person with high body fat has high leptin levels, objectively high, yet their brain behaves as if leptin is absent. The hunger signals continue. The energy conservation signals continue. The fat tissue is screaming at the brain, and the brain has learned to not hear it. Research from Harvard, led by Professor Manzoros and his group using functional MRI, has been specifically studying which brain regions activate when people are shown food cues under conditions of varying leptin sensitivity. The findings confirm that leptin resistance doesn't just affect appetite signaling, it affects how the entire reward system responds to food-related stimuli, making high-calorie foods appear more desirable and harder to resist at the neural level. Now let's talk about what happened in 2025 that genuinely changed our understanding of this entire system. A study published in Nature Metabolism, reviewed in a commentary titled Signaling from Adipose Tissue to the Brain is Not Just About Leptin, documented a previously unknown pathway with striking implications. Under acute psychological stress, fat cells in white adipose tissue undergo lipolysis. They begin breaking down their stored triglycerides into free fatty acids. Those free fatty acids trigger the release of a molecule called GDF-15 from local macrophages in the fat tissue. GDF-15 then travels through the bloodstream to the brain, where it binds to a specific receptor called GFRAL, located in the brainstem, and the documented effect of this GDF-15-GFRAL binding is the induction of anxiety behavior. Let that sink in for a moment. When you experience acute stress, your fat tissue breaks down, releases a chemical signal, and that signal travels to your brain and makes you feel more anxious. Your fat is not just responding to your stress, it is amplifying it, feeding it back into the brain through a dedicated molecular pathway that, until 2025, nobody had identified at this level of precision. This is not a trivial finding. It establishes a direct bidirectional relationship between your adipose tissue and your psychological state, a relationship that goes far beyond the long understood role of cortisol in stress-related fat accumulation. The fat itself is an active participant in the stress response, not merely a passive downstream consequence of it. But the story of fat and the brain is not only about stress. Let's talk about adiponectin and why what it does for your brain is genuinely remarkable. Adiponectin is another hormone produced by fat cells. Unlike leptin, which rises with fat accumulation, adiponectin behaves counterintuitively. It is actually lower in people with higher body fat. Lean individuals tend to have higher adiponectin levels, and the research on what adiponectin does in the brain is striking. The 2026 Obesities Review documented that adiponectin directly modulates synaptic plasticity and neurogenesis, the brain's capacity to form new neural connections and generate new neurons, particularly in the hippocampus, the region most critical for memory and learning. In animal research, adiponectin administration has been shown to promote hippocampal neurogenesis. In human studies, lower adiponectin levels are consistently associated with worse cognitive performance and increased risk of cognitive decline. An elevated adiponectin has been associated in multiple longitudinal studies with better preserved cognitive function across aging. This means that the state of your fat tissue, specifically how much adiponectin it is producing and releasing, is directly influencing the neuroplasticity and memory function of your brain, not metaphorically, through documented molecular mechanisms. A third adipokine worth understanding is irisin. Irisin is particularly interesting because it appears to be one of the molecular links between exercise and brain health. When muscle and fat tissue are activated by physical exercise, they produce irisin, which crosses the blood-brain barrier and has been documented to promote BDNF, brain-derived neurotrophic factor, the same protein we have discussed as critical for hippocampal health and cognitive resilience. This means part of the brain benefit of exercise is mediated by fat and muscle tissue producing a hormone, irisin, that then travels to the brain and stimulates its own growth factors. Your fat, when healthy and active, participates in protecting your brain through exercise-induced hormonal signaling, and the clinical implications of all of this are now coming into sharp focus. A growing body of evidence, including findings highlighted in the 2026 Obesities Review, identifies adipokine dysfunction, AS, specifically leptin resistance and low adiponectin tiers, as potential biomarkers for early cognitive impairment risk. Researchers are actively exploring whether adipokine profiles could serve as early warning signals for Alzheimer's risk years or even decades before symptoms appear. The adipose tissue-brain axis is no longer a peripheral curiosity in neuroscience. It is becoming a central focus of research into cognitive aging and dementia prevention. So what does all of this mean practically? The evidence points clearly toward maintaining metabolic health, not just for cardiovascular or aesthetic reasons, but specifically for the brain communication that depends on healthy adipose tissue function. Chronic excess body fat disrupts leptin signaling, producing leptin resistance that impairs the brain's ability to regulate appetite and increases reward sensitivity to high-calorie food. It reduces adiponectin levels, removing a documented promoter of hippocampal neurogenesis. And the 2025 Nature Metabolism discovery suggests it may amplify the brain's anxiety response to stress through the GDF15 pathway. Exercise specifically addresses multiple parts of this system simultaneously. It improves leptin sensitivity, increases adiponectin, and generates irisin, all of which benefit the adipose brain communication system directly. Dietary quality matters for exactly the same reasons it matters for every other system we have discussed. Chronic inflammation from ultra-processed food impairs adipokine function and worsens leptin resistance, while anti-inflammatory dietary patterns support healthier adipokine profiles. And sleep. Leptin is strongly influenced by sleep. Sleep deprivation measurably reduces leptin and increases ghrelin-SPA, the hunger hormone, which is why a single night of poor sleep reliably increases appetite the following day. Protecting your sleep is protecting the hormonal communication your fat tissue sends to your brain. Let me bring everything together. Your fat is not silent. It is not passive. It is an endocrine organ, classified as such in peer-reviewed literature as of 2026, producing dozens of signaling molecules that cross your blood-brain barrier and actively regulate hunger, mood, stress, memory, and cognitive health. The 2025 Nature Metabolism study showed that your fat amplifies your anxiety under stress through a dedicated molecular pathway nobody knew existed until recently. The 2026 Obesities Review documented that adiponectin from your fat directly modulates the neuroplasticity and neurogenesis of your hippocampus, and leptin resistance and the breakdown of fat-to-brain communication that occurs with chronic excess fatty impairs the hunger signaling and reward sensitivity of your brain in ways that compound the very conditions driving it. Your fat is talking to your brain every single hour of every single day. Whether that conversation is helping your brain or hurting it depends almost entirely on the metabolic health of the tissue producing the signal. The real question was never just, how much do I weigh? The real question is, what is your fat tissue currently saying to your brain, and is it helping or working against you? If this completely changed how you think about body composition and brain health, subscribe, because next week we go even deeper into the biology connecting your body to your mind. See you in the next one.